This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 348, recorded on July 31st, 2015. Hey everybody, I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today from somewhere in Oregon, Rich Condit.
Howdy there, Vincent. How are you?
I'm very well. It's gorgeous here today. We got
that's good. What do you got?
Puffy clouds, mostly blue sky, 30 degrees Celsius. You know, we have had real summer this week. We've been in the mid-30s and lots of humidity. Today we have 37%. But I love that hot and humid weather. I should have been living in Gainesville, I guess. In Gainesville, you get that in spades. So what'd you say, 30 Celsius? Yeah.
So that's about 85, 86 Fahrenheit. Okay. I have to do that. You know, I'm getting close.
That's right.
Well, somewhere, you said I was somewhere in Oregon. In fact, I'm in a place called Sun River, Oregon, a little south, about 20 minutes south of Bend by car on Highway 97. I'm here for, of course, this is our great circumnavigate the U.S. road trip boondoggle.
Mm-hmm.
And my wife's family has a vacation house here in Sun River, which is a sort of a resort community that they built like 25 years ago. And so members of the family can come and hang out here when they want. I think we've been here for a week. I think we'll be here for probably 2 more.
Nice. Before
we continue on. And it's gorgeous. It is 88 degrees Fahrenheit. That is 31 Celsius. And it's like probably, I need to look this up, it's probably about 35% humidity. So this is high desert. Holy cow, 16% humidity.
That's very low, yes.
The dew point is 36 degrees. Last night the temperature went down to about 50. It's supposed to go up to 96 today.
Wow.
And my guess is that when it gets there, it'll be down around 10% humidity or lower. So it's interesting weather because it's high desert. We get a large variation in temperature range, but it's been very pretty. And this is right. It's basically in the middle of the Deschutes National Forest, and the house overlooks a river and a lot of bike paths around. And we also 2 of my wife's sisters are in town, so we have a good time. Great.
Sure. Sounds good.
Yep. Great fun.
Well, it's you and me, Rich, because Kathy and Dixon are on vacation, and Alan had some car problems.
There's just no excuse.
I don't know why.
There's no excuse.
You don't need a car for TWiV, right?
No. And what's this about vacation?
Yeah,
right?
Really. I'm
on permanent vacation, and here I am on TWiV, okay?
Well, thank you. I don't know if you and I have ever done one together, have we?
I think if we look back through, we would find one where you and I have been doing this solo. Because, for a long time, it was you and me. For a long time, it was you and Alan.
That's right.
It was you and Dixon.
Right.
And then you and Alan and Dixon. And Dixon kind of goes in and out, so it's you and Alan.
Yeah, that's right.
Then it was you and me and Alan. And I think there were a couple of times one or two times when Alan couldn't make it.
I guess that's a phenotype of August, right?
Yep.
But I'm here. I'll do TWiV every Friday,
There you go.
I love it.
So do I. And I will, despite my protestations to the contrary, I understand being absent now and then. And there may be a week or 2 up here in this road trip where it doesn't happen. But anytime I can possibly make it, I'm here. It's great fun. All
right. Actually, I will be unable to record on the 14th of August because I'll be in Washington interviewing a microbial artist, and so I will release a canned episode on that day, but it'll be a good canned episode. Yeah.
Actually, that's probably one of the days that I would have trouble making it anyway. So 14th. That works fine.
Yep.
Okay.
This episode.
Wait.
Yes, sir.
Microbial artist. What's that about?
Microbial artist. So you may know Michelle Banks. She has a small company that makes watercolors and other representations of microbes, bacteria, and viruses. My desktop on my computer is by her, and she's got a shop on Etsy. Let's see, at ETSY Michelle.
I think we picked this before, right?
Yeah, it was definitely a pick of the week. Yeah, Etsy.com/Artologica, ARTOLOGICA. That's her store. She makes scarves, watercolors, all sorts of cool stuff, and I've bought a bunch of it. So I'm gonna. She lives in Washington D.C., and I have for a long time wanted to interview her about why and what she does, right? So I finally talked ASM into giving me a camera crew, and I'm going to go out and chat with her, and I'll release the video as a TWiV special.
Excellent.
And because I think it won't work as an audio only, because we're going to be watching her do her work, right? So,
right.
But it'll be a video, and it'll be in the TWiV. It'll be on the TWiV channel on YouTube. So that's August 14th going up. The 16th. Actually, no, it'll go up later, because I'm going to release something else that weekend, because we won't have time to process it by the 16th.
You said Artologica?
Yeah, ARTOLOGICA. Etsy.com/Artologica. You're not getting it?
No.
Etsy.com/ARTOLOGICA. Yeah, you're right. Let me give you a better URL. Michelle Banks Artologica. Oh, okay. That's because it's Etsy.com/shop/Artologica.
Okay.
Mitochondria, chromosomes, brains, bacteria, viruses. She even makes Christmas tree ornaments of Petri dishes. All kinds of cool stuff. Atrial fibrillation. Yeah, you're probably looking at it now, right?
I'm just really slow with my fingers here. I've almost got it. I got a dot instead of a slash. You know.
You're going to end up just. Well, I'll tell you what. While I
got it.
While
I'm
I
here.
do this first ad,
I've seen this before. Yeah, you do the ad. I'll look at what she's got.
This episode of TWiV is brought to you by ASM Gap. They are running a conference called Turning Your Science into a Company. This is taking place on October 1st through 3rd, and you need to apply to get in. The deadline is August 20th, which is more or less 20 days away. And if you've ever had an idea from your science and you've wanted to turn it into a company, you should consider going to this meeting. It's basically a workshop. It's going to be held at ASM headquarters, which is in Washington. 2.5 days. Provides basic information on starting your startup, and they will use examples from companies who have already done it, biotechnology industry. Day 1, planning a startup. Day 2, executing the plan. Day 3, strategically exiting. In other words, selling it and making a lot of money. This will be directed at advanced graduate students, postdocs, and early career scientists, and you will get tips, advice, and resources from people who have done this, people who have started up. People with a lot of experience in doing this. This sounds great to me. I would love to be able to do this.
It sounds really interesting. Sounds really interesting.
So
I've got Michelle Banks. I'm sorry. You done?
Yeah. Let me just give out the URL. ASM Gap. That's one word. .org/TSC. Application deadline August 20th. Check it out. All right. Rich.
So I got her site here. It looks very cool, and I'm going to say no more because you're going to do a whole episode on her, and I will listen in rapture.
Yeah,
I'm very interested to know what motivates her.
Yeah, for sure.
And I hope she's actually making a living doing this, because I really like, to support efforts like this. This is great.
Yeah, it's one of the things I will ask her. Is it a hobby or a profession?
Right, probably somewhere in between.
Yeah, I'm suspecting, yes. I have another job announcement. Well, no, not another. I have another announcement. This is actually a second job position at the NIH. Last week, we talked about one at the Laboratory of Infectious Diseases. This is another virology tenure-track position in the HIV Dynamics and Replication Program, which is part of the National Cancer Institute of the NIH. They want a tenure-track investigator to work on an area of virology, including but not limited to HIV retrovirus biology, similar to a position equivalent of assistant professor in a university setting. They want people with expertise in HIV, retrovirus biology, or cancer virology, virus replication, virus host cell interactions, drug resistance. You need to have a PhD or equivalent, some postdoc experience, and a record of high achievement. So these they're going to start reviewing applications on August 3rd, which is Monday. But if you hear this and you're interested, write, because they will keep reviewing until the position is filled. And I'll post a link to the job in the TWiV show notes. So you have to go there to check it out. So now we have 2 weeks in a row, 2 virology positions available at NIH. So
you suppose they'll get any applications for those?
I think they will get lots, yeah. This one is rather limited. You know, they want HIV retrovirus, but there are lots of those individuals out there. Mm-hmm. I also want to mention to our listeners a contest that we have mentioned before. This is the Agar Art Contest, which is being sponsored by ASM. It's very simple. You create a piece of agar art. You take a plate, a plastic petri dish with agar in it, and you figure out how to make some art on it using microbes to grow and make the art. And the deadline is September 3rd. I will put a link to this in the show notes, but you should enter because a lot of our listeners have a lab and they can get petri dishes with nutrient-rich agar in them. We have them too. So check this out. And one of the judges will be Michelle Banks, who we just talked about. Okay. Yeah. So please submit something. We'd like you to do that.
Outstanding. No gain of function stuff. Okay. That's not allowed.
Right. We have some follow-up from last week's episode. We did a paper on rosette virus, and the first author, Alma, writes to us. Thank you for mentioning my JGV paper and one of my favorite plant viruses. I didn't pick the title, and I would have gone with something less flashy if it had been up to me.
That was that title that said something like 70 years in the making or something like that?
Yeah.
You had some objection to that.
Yeah, I don't mean personally, of course. I just thought it was a little flashy, and Alma agrees. Okay. You mentioned in the episode that you weren't sure where the name Amaravirus came from. The name Amaravirus comes from the first letters in the name of the type species of the genus, European mountain ash ring spot associated virus. Not very creative, but it goes with the descriptive nature of naming plant viruses. I think it's very creative. I like these namings.
I'll tell you later what all plant viruses are called.
Okay.
I can't tell you. It's not suitable for our family audience.
Now, last week...
Remind me.
Okay, last week, along with my pick, this was a pick having to do with how many people consume podcasts on iPhones versus Android devices. So I decided to put a poll up for TWiV listeners. I asked, how do you listen to TWiV? We had 89 replies. 35% use an iPhone, 22% use an Android, 26% on a laptop or desktop, and 15% other.
What's other?
I don't know. Maybe like a tablet. I forgot to put tablet on. Maybe a lot of those are tablets, or maybe on a TV. You can listen to them through TV in a way. You know, I don't know.
Suppose.
I didn't tell them to drill down, but so that's a little less than the poll. The poll said 80% consumption of podcasts on iPhones. So,
well, I'll make it 90 here. So I listened to mine on an iPhone.
All right. Steve writes, "Hi, Vincent. I listened to your latest TWiM the other day. I've caught up with them all now, and thought you sounded not your usual bright and breezy self. Voice was lower and slower. Had me a little concerned. Glad to say it must have been some recording difference because you were all back to normal today on TWiV. And this squirrel story was quite an education in how difficult it is to find something even when you're sure it must be there. Do look after yourself. You'd be a hard act to follow." Oh, man. I'm feeling fine. I'm in that TWiM, I was fine. I don't know why the voice was off, but there was nothing wrong with me. I was okay. When I do podcasts, I'm really happy because I love them. And I know I'm talking to people who want to learn about microbiology. Steve is from England, and he says, where my central heating has come on, and it's rained all weekend, the first blackberries are ripe already. I hate seeing that, as I'm sure they used to wait till autumn, and it makes me feel like summer is over. Well, tomorrow's August 1st, and when August hits, that's it.
Is that right?
Well, college starts in August, right?
Yeah, I guess so.
Like in a couple of weeks, they start going back to school.
Oh, that's scary.
You want to take the next one, Rich?
Sure. Chaim, is that how I pronounce that?
Chaim.
Chaim?
Yep.
Chaim?
Yep.
Chaim writes, can't just fire a graduate student? Hopefully true at Columbia, but definitely not everywhere. I was fired from my first PhD lab with no warning about 2.5 years in. The graduate program told me there was nothing they could do for me unless I found another PI with available funding who was willing to take me on. Fortunately, I was able to do so, though it meant completely switching fields. I am fully aware of how incredibly lucky I was, though, and most people in that situation would have ended up on the street. This is in response to a communication we had last week where the mention was made of the notion of firing a graduate student, and we said, you can't do that. Now, actually, I will say that in my experience, entering a program as a PhD student is not necessarily a free ride to the end. One has a mentor, one has a supervisory committee, a committee of individuals who monitor your progress, and hopefully the supervisory committee functions in a fashion to moderate any craziness that might come out of the mentor. But their function, in addition to advising you, is to monitor your progress. And students, even after passing their qualifying exams and stuff like that, can, not live up to the expectations of the mentor and the supervisory committee, and can be, dismissed from a program through a formal means. And under those circumstances, the graduate school, doesn't, you can appeal to the graduate school, but they don't necessarily have anything to do with it. Sometimes, you could find another lab. I'm not saying that this is what happened to Chaim, but those are the circumstances that I know of where a PhD student can be let go. Now, I don't know of any circumstances where a mentor just says, you're out, you're fired, and there's no recourse for appeal, but we have someone here who says that has happened. So yeah, my experience has been that it's not a permanent position, but there is a process associated with letting go of a graduate student. Mm-hmm. It is not a whimsical decision, right? And as the letter last week implied, it is not something that you use to make someone productive, to threaten to fire them. If it happens, and I wouldn't be surprised if somewhere that happens, right? Oh, yeah. But it should not. This is an academic position, and, if you don't perform, if you're academically in trouble, you can be let go, but there's a process you have to go through. You have to document the issues. Now, of course, if you run out of money, here at Columbia, if a PI runs out of money, the department will take care of that student, put them somewhere else, and not the onus is not on the student to find somewhere else to go. But again, that may not be the same everywhere else. That's certainly been my experience at two other institutions, and that is that when a program takes on a student as a PhD student, they make a commitment to, as long as the student is in good academic standing, to see that student through to the end, despite whatever financial or other difficulties the mentor or the mentor's lab might run into. And there are circumstances when the mentor turns out to be a crazy or something like that, and the department will take care of that too. Help work it out. All right. I meant to say to Steve, the previous letter, thank you for your concern. I do appreciate it. I forgot to say that, and that's very important. The next one is from Ted, who writes, Professor Racaniello, thanks for reading my email about Jokichi Takamine, whose remarkable life I encountered several years ago
while researching the history of local anesthesia. This was an email we read last time about this individual who donated cherry trees to 2 different places, one of which was Washington, D.C., and the other here in New York City. Mea culpa, his crypt is located in Woodlawn Cemetery, Bronx, New York, which is a National Historic Landmark, not Woodside Cemetery, which I had written. Just wanted to make this factual correction. Sakura Park, a pocket park, is located at 500 Riverside Drive at 122nd Street and is sandwiched between Riverside Church on the south, Manhattan School of Music on the east, and Grant's Tomb on the west. Still possibly not a safe place to venture. In any event, thanks for being such a great podcast host and good sport. I admire your energy and breadth of knowledge.
Ted still hasn't told us what he does. Because I was wondering that last time because he had a remarkable essay on this guy. It was great.
Yeah, he didn't have his position in the last letter? I thought he had.
No, I don't think so.
No? Okay. You can take the next one from John, please.
Wait a minute. I want to know if you have not seen these cherry trees in New York, right?
I haven't gone looking for them, no. But they're not going to be in blossom by now, right? It's too late.
Right, no, that's like April. So would this not be a safe place to venture?
So 500 Riverside is right near Grant's Tomb. It's okay. It's slightly north of Columbia University. I've been past it many times.
That's the undergraduate campus, Columbia University.
In fact, the university has a place called International House, which is a residence for international students when they first come here and they're looking for a place to stay. They can stay there. And it's about 120, I think, or maybe 118. So this is okay. You know, sometimes I go by Grant's Tomb and he's sitting out there catching rays.
Well, next spring, you need to go by there and take a picture of the cherry trees, and we'll use them as an episode picture.
All right, I'll do that. No problem.
My turn?
Yep.
John writes, reading my letter about insects on TWiV 347, you asked what I do for a living. Oh, maybe this is
that's the one you're thinking about, yeah.
Okay. Insects are just a hobby. I write CAD software for a living. Cool. Really cool. One paper on TWiV 347 was about squirrel-associated virus. You might say squirrels are a hobby too, but I don't breed them. You read my letter about squirrel-borne virus on TWiV 329. This guy is a Renaissance man. I want to repeat my observation from that letter. Scurus variegatoides is common near people in Central America, like Scurius carolinensis in, pardon me for butchering these names, in eastern North America. If it were a bubbling cauldron of deadly virus, we would see more disease. Tourists would be dropping dead. Dogs would be saying help instead of squirrel. This reminds me of, what's the herpes virus? B?
Yes.
That hangs out in monkeys?
Yes, right.
That is so deadly in humans. And this was in spillover, I think.
I think so, yeah.
They did epidemiological studies because it's very widespread in monkeys, and there's lots of places where these monkeys are right up close to humans, but few, if any, I think, know documented cases of transmission of the virus to humans. There are only laboratory cases where people have, stuck themselves or whatever and gotten very sick. So this reminds me of that. The species was discussed in Mammalian Species 500 and, of course, in Squirrels of the World. And he gives a couple of links. P.S. Your discussion of the difference between male and female Legos at the end of TWiV 343 reminded me that when I was a kid, my sister and I undressed her Barbie dolls. Poor Ken.
Yes, really, poor Ken.
Okay.
So John he had written in 329 about this very report. It wasn't published yet. It was a report in a newspaper, I believe, a news site. So he was on it. And then it came out in the journal, and we did it. So I should have remembered, but I didn't. Thank you, John, for pointing that out. And he's right. If this were a big problem, we'd have many more cases.
Right.
So it's pretty rare. The previous author, Ted, is a professor in a dental school.
That's right.
So that's what he does.
Okay, yes. That's right. We had that. Okay. We're all straightened out.
All straightened out. All right, let's do a couple of snippets here. I was looking for papers on Monday, and I came across this one, which I didn't think was a full discussion, but Merrick's telling our listeners about. It's a paper published in PLOS Biology. It's called The Discovery, Distribution, and Evolution of Viruses Associated with Drosophila melanogaster. And it is from the UK, University of Edinburgh, Cornell University, Department of Entomology, and the University of Ghana.
First author is, Claire Webster. Last author is Darren Obard, and it has, pardon me for the et al., but quite a few authors in between. Well, Waldron, Robertson, Croson, Ferrari, Quintana, Bruqui, Bain, Longdon, Buck, Lazaro, Corley, and Hadrill.
So this paper is neat because they looked in wild Drosophila. As many of you may know, Drosophila is a laboratory subject. It's been very important for understanding genetics and development, and so many labs study it and work on it. And they went. But
if you take a peach and stick it out on your picnic table for a while,
that's right,
it will be visited by one of these creatures.
That's
little
right.
teeny flies, kind of reddish, maybe. That's the way I think of them, though there's probably all different colors.
Fruit flies, right.
Yeah.
So they went out and collected a whole bunch, over 2,000 from Kenya, from Ithaca, New York, and they ground them up and made total RNA. And these are all different species of Drosophila. And they did RNA sequencing to find out what kinds of viral sequences were in there.
In the process, if they've done several different things here, but I think in this pooled one, they gathered pools of flies, but they did sort them out morphologically. They weeded out the ones that didn't look like Drosophila, so that they were focusing on Drosophila, because there's a bunch of different critters that will get trapped with the baits that they used.
Right. So they found a number of known viruses of Drosophila, and they
known because they've been seen in laboratory strains of Drosophila.
Seen just laboratory, and many people work on these, Drosophila C virus is one of them. And then they found lots of new viruses. What they say is that over 30% of wild Drosophila melanogaster, which is the strain that the species used in the laboratory, carry a detectable virus, and more than 6% have multiple viruses. They also it's an interesting approach they use to identify new viruses, right? Because you look at these sequences and they're not in the database. So how do you know they're a virus? Well, they found virus-derived small interfering RNAs in these flies. They're derived from the viral genome, but they're 21 to 22 nucleotides long, so you know that they're being processed by Dicer in the insect. So that's how they identify the larger sequences as viral. That's pretty cool.
Right. They actually, I think they took a couple of approaches. That was one of them, which I thought was quite ingenious. And I think they had what they call blast hits as well. So I think a lot of these sequences picked up stuff that looked viral,
right,
in the database, but wasn't necessarily. Well, certainly the new sequences they had were not recognizable as viruses that had been deposited in the database before. So, both by homology to existing virus sequences and by this siRNA trick, which is quite clever.
Yeah, very cool. The other neat thing is, all right, so the community of viruses that you find in laboratory strains, which has been characterized before, is quite different from the wild collection of viruses. But some of the new viruses are also found in laboratory lines. Mm-hmm. And they're also found in cell cultures derived from Drosophila, which many laboratories use. And the other thing I wanted to point out is that a lot of these Drosophila have Wolbachia in them, which is a symbiotic bacterium that's an intracellular bacterium. It lives within insects, and like at least 40% of arthropods in the world have Wolbachia in them. It's thought, and it's known that Wolbachia confers resistance to some viral infections. And in fact, we've talked here on TWiV about attempts to use that property to make, say, dengue-resistant mosquitoes. But they were not able to make any relationships between the presence of Wolbachia and the presence of any virus. Right, so there's no correlation. If you have Wolbachia there's no virus that's particularly absent, which is kind of interesting.
Yeah, I think that's quite interesting.
I think that these are viruses of Drosophila, and maybe they have adapted to the Wolbachia, whereas, what we do is we take a mosquito, we infect it with a Wolbachia, it becomes resistant to a human virus that's vectored in the mosquito. And so that's a new thing for the virus to encounter in the mosquito. So that may be why they're inhibitory, but not these naturally occurring viruses. Anyway, that's
so they found the total catalog here.
Yep.
Is like 21 new viruses.
Yeah.
And 20 of them are RNA viruses. There's only one DNA virus, and that guy is a double-stranded DNA virus that looks like a nudie which is an insect double-stranded DNA virus, fairly large dude.
Yep.
And all of the rest of the RNA viruses include, most of what you can think of: positive strand, negative strand, double strand, everything.
Yep.
So that's interesting.
And they give them provisional names. I think some of these they name them after where they found them. Mm-hmm. Berkeley virus, Bloomfield, Brandeis virus.
La Jolla virus.
La Jolla virus, there you go. Torrey Pines
virus, a little more specific.
It's pretty funny. Anyway, it's pretty neat. It's good stuff.
Yeah. I'm struck by, this is another example of the power of not just sequencing, but bioinformatics. I'm really impressed when I read these papers now, how creatively one can use sequence information to address biological questions. And that's where this, for example, the siRNA thing comes in, because they make a big point of discriminating between the RNAs that are short enough to be dicer produced as opposed to other small RNAs that can be found in the same cells, to focus in on which ones might represent a response to an active viral infection.
Right.
So very interesting.
Dennis wrote in with a story that I think is worthy of a snippet. Doc, I just saw this on the BBC News app and thought you should see it. Nigeria marks 1 year without recorded polio case. So I went over to polioeradication.org. No wild poliovirus type 1 cases reported in the past week, and none have been reported in 2015 in Nigeria. They had 6 cases in 2014. The most recent one was on July 24th, 2014. Now, there is vaccine-derived poliovirus in Nigeria. There has been 1 case so far this year caused by vaccine-derived type 2. Okay, so the Nigeria being free is free of wild-type polio. Mm-hmm. So on July 24th, which was last week, it was 1 year since the last case. And if the country goes 2 more years, then the WHO will consider declaring polio eradicated from Nigeria, along with the rest of the African region. So that, Nigeria is the only country in Africa where the virus has not yet been eradicated. So if it can be eradicated there, then African region will be declared eradication of polio. But of course, they have to keep it clean for 2 more years. So it's not so easy because it can come back.
Right. This link that I guess you put in here to the nature piece is really a good read. That's I really enjoyed that because it, among other things, emphasizes how important the vaccination campaign and the vigilance is in making this happen, and it emphasizes, what the barriers to eradication are, them being mostly social and political. And this one quote from that article really grabbed my attention, because one of the problems has been Boko Haram interfering with vaccination. And it says, this is a quote from this news piece. In areas in the northeast that were controlled by Boko Haram, health officials mounted a quote hit and run campaigns, rapidly vaccinating children on short notice when security conditions allowed. And I'm picturing this, and I'm thinking how dedicated these people are to go in and do that, because they're risking their lives.
Yep.
It's amazing.
What I find really nice about this, that they're free for a year of polio, is that as recently as 2003, Nigeria stopped immunizing its population because they were suspicious of the vaccine. Mm-hmm. And that led to a huge outbreak of polio in the country. It spread to other countries in Africa that had been free of polio. This was a real problem. They have come around. They have embraced immunization. And in fact, they are now using Salk IPV, as recommended by WHO. It has to be injected. So they're doing the good thing. I'm really impressed.
And according to this article, one of the reasons that they came around was that the neighboring countries into which this was spilling over finally said, hey, dudes, get your act together, okay? You're hurting us. And so, there's large-scale social pressure to get this done. Very interesting.
You had a snippet.
Yeah, so I've been corresponding with our old buddy, Tyler Sharp. And for those newer TWiV listeners, I would refer you to TWiV 178, entitled, this is typical Alan Dove, T-Sharp on how tequila mosquito. In order to get that whole thing, you have to listen to the episode. But Tyler, at the time that we interviewed him, was an officer in the Epidemiologic Intelligence Service for CDC. This is CDC's, I believe, 2-year fellowship, right. The members of which comprise their sort of epidemic SWAT team that goes out and chases down and tries to control epidemics of all sorts, not just infectious diseases. And so Tyler was an officer in that and has since established himself in Puerto Rico as a CDC officer running, I believe, their dengue surveillance and operation there. But he still gets called up occasionally to go out on assignment, and he's gone out on one recently. I chose this link here as a snippet, which is when he went on I forget where the mission was that he blogged about previously. But he occasionally blogs about these, and then they, of course, this is CDC, so the blogs have to be posted, so have to be cleared by and posted through CDC. The link I've got here that we can turn our listeners on to is a blog that he wrote about dengue in Africa. Apparently, there's a larger burden of dengue in Africa that people have been aware of. Primarily because it gets quite often misdiagnosed as malaria. And so the blog and other things are an effort to sort of correct that situation, because proper diagnosis leads to better treatment, which leads to better control. But more recently, and this is the correspondence I've had with him, he's on a mission to American Samoa. Actually, he's coming back today. There was an outbreak there that, let me see, I've got the last figures from him here. The last official figures were 184 cases with 4 confirmed dengue deaths. I had to look up American Samoa. It's out in the middle of the Pacific. The closest large landmass is New Zealand. I don't know what the population is, but it can't be very big. But at any rate, they had this outbreak there, and so Tyler headed a team of 6 individuals that went out and took a bunch of public health measures that consist of, teaching people how to clean up the mosquitoes, doing epidemiologic surveys to find out how bad the epidemic really is. Most importantly, educating people on reporting and coming into the clinic with cases in their earliest stages, because proper treatment, as we know, consistent with the Ebola epidemic, really is assisted by getting people in an appropriate clinical situation as soon as possible. I had some questions for him because there's a fairly high case fatality rate here. It's up to 3%, which is one of the reasons they got called. I mean, it's not at this point a big outbreak. 184 cases, 4 confirmed deaths, but you can imagine when this comes up, you don't know what's going to happen. And so getting a CDC or some sort of team on it right away makes sense, and it sounds like they've been relatively successful. But I was curious with that sort of a case fatality rate, as we've discussed in the past, the fact that dengue is in 4 serotypes. If you are infected with just one serotype, you can have anything from a fairly mild to a fairly severe disease. But one of the biggest problems comes if you have been previously exposed to one serotype and then you get exposed to a second serotype. There is an enhancement of disease that gives you the severe hemorrhagic disease. So my question to him was, what serotype are you dealing with, and has there been dengue there before? And his response was that it's dengue 3, and the island population is 96% seropositive for dengue.
Wow.
As is not untypical
Yes.
in the tropics. And they've had several epidemics through before, so these people have been exposed really widespread to other serotypes, so it's not a big surprise that should any given serotype come through, they're going to see a lot of dengue hemorrhagic fever.
Yep.
So, good work, Tyler.
Nice.
It's been fun corresponding with him, and I'm glad to see him doing a good job in American Samoa.
It's cool. Tyler's a light colonel.
Yeah.
That's cool.
He's in his uniform.
Yeah.
And in a lot of these communications I get from him.
All right, we have a paper to tell you about today, and I have to say this is a paper in PLOS Biology. It's called Imperfect Vaccination Can Enhance the Transmission of Highly Virulent Pathogens. The authors are Reed, Balgent, Powers, Gosana, Blackwell, Smith, Kennedy, Walkden, Brown, and Nair, and it's from Penn State University, NIH, the Purbyte Institute in the UK, and the University of New England in Australia. I received this last week from a science writer in the UK by the name of Ed Young. He gave it to me under embargo and wanted some comments on it. So I've been looking over this for a while, and as soon as I got it, I said, this is for TWiV.
And also, as you know, my wife saw one of the news things on it and sent it to me, which I then forwarded to you. Right. So this has been making the rounds.
Yeah, this has been in the news, and so I want to go over the science a bit. I just want to preface it by saying that I think the science in this paper, the experiments are well done. I think it's overinterpreted, and I think the vocabulary they use is really not appropriate. And just in the title, imperfect vaccination, and they also call these vaccines that we'll talk about in a moment leaky vaccines. I object to the use of words that imply a defect. Because there's, as you will see, there's no defect here. But even more than that, and I think Rich will agree, he has some comments as well, the way it's presented is very odd because the data are clean for what they're addressing, but the way they're presented is a bit stretching. Anyway, this is about this hypothesis that if you had a vaccine that prevented disease but did not prevent virus replication and shedding and presumably transmission, would that kind of vaccine enable the selection of more virulent viruses? The idea being, let's say a virulent virus kills you really quickly, you don't have time to transmit it, so those viruses eventually are selected out of the population.
Yeah, I think of them as burning out.
They burn out.
Okay,
yeah.
They're so hot that they kill their host really rapidly, and so they kill the thing they need to transmit. They burn out, and it's been a long-standing hypothesis that viruses, in fact, don't do that they reach an equilibrium with their host so that they can maintain a chain of transmission. But when we're talking about vaccination, we're talking about introducing artificial situations into a population.
Right. So if you had a vaccine that allowed transmission, you could imagine that a virulent virus could perpetuate. And
right.
Okay,
so if you had ordinarily a virus that just kills things and would burn itself out. But now you introduce a vaccine that allows those hosts to live but still transmit the virus. It might permit the continued transmission and therefore the perpetuation of that virulent strain rather than it burning out. You let the host survive, so you get rid of that burnout thing.
Right. So this is the hypothesis, and just to start, I think it's flawed because of a number of reasons. I mean, first of all, you can imagine that transmission could be enhanced without becoming more virulent. So you could, in these individuals who are immunized, they allow virus replication but not disease. You could see maybe transmission increasing without virulence increasing. But I also think that, I mean, I'm trying to think of how much more quickly do you have to kill someone. You know, I don't know how quickly Ebola can kill you, for example. It's going to take, you have a 3 to 21 day incubation period. And let's say in some cases you get killed in a day. Well, we know it can still spread. I mean, that's pretty rapid, but it still spreads because people take care of you and they get infected and so forth. So I'm not sure of how much more quickly. I mean, in this paper we'll see an example of this experimentally, but you know, in the wild there are lots of viruses with a wide range of lethality times, and I'm just not sure that, you know, this is a good selection feature. I do think there are lots of other selection parameters out there that can act on viruses and help them to evolve, but I'm just not sure that this is one of them. The other thing is that we tend to, as humans, we tend to think that better is always the goal of evolution, right? Because humans always want a better car and a better building, you know, a better razor or whatever. And it's not the way viruses work. The viruses in a host, all they have to do is get through that host to the next one. They're not, selection doesn't try and build something that's going to be good in 10 years or in 20 years. It selects those that can't replicate in the host, and the ones that can go on. So I'm not sure that this idea of increasing virulence and transmission, I just think it's too simplistic. You know, there are lots of selective forces in a host and individual cells in the environment that are working on viruses. To just put it towards virulence, I think, is wrong. So those are my objections, and I've been thinking about this a long time, and that's my fundamental objection to the hypothesis.
Yeah, I think that to me, this is kind of an experiment in a very basic concept.
Yes.
Okay, and that is that, you know, if your hypothesis is that, and you know, strictly conceptually, if you're without necessarily thinking about what actually might go on in a natural situation, if your hypothesis is that if you somehow through vaccination extend the life of a host that would otherwise be burned out by a highly virulent pathogen, that you would promote the transmission of that highly virulent pathogen. As a fundamental hypothesis, that's it's a perfectly reasonable, what if idea, and a perfectly reasonable experiment to do. And the experiments are nicely constructed to test that hypothesis. The issue becomes, what does this really mean in the natural world? And that's where all of these other questions actually come in.
All right, so the experimental system they use here is chickens. And chickens, as you know, we grow lots of them to eat and to get their eggs and so forth. So we have a huge poultry industry globally. And
actually, I'm reading a book called Sapiens now about the development of the whole history of, this will be a TWiV pick if I can ever get through it. Just to cut to the chase, there's 25 billion chickens on the planet.
Oh, so those are bred by humans, right?
Yeah, it's more, from that point of view, the chickens are a very, very successful species because they've conned humans into breeding them.
That's
more
great.
Successful than any other species on the planet.
Wow.
Any other animal species.
So for years, it's been known that herpes virus, called Merrick's disease virus, will infect chickens and spread among flocks and cause disease. And this is not good, of course, because you cannot use a diseased chicken.
Did you say this was a herpes virus?
I said it was a herpes virus, right? Marek's disease, herpes virus. So at the end of the 60s, vaccines were deployed to use to immunize chickens so that they don't get disease from this virus, and they can be used.
And I'll have more to say about this later on, but at this point, I think it's at least interesting to point out what the vaccines are. They're live vaccines.
Right.
And from my understanding is the first two were actually turkey herpes viruses that were not modified in any way, but since they're going outside of their normal host range, they can infect these birds but not kill, but there's cross-reactivity to the Merrick's disease virus, so the birds are protected. This is precisely analogous to using cowpox or horsepox to vaccinate against smallpox.
Right.
Using a pathogen from nature that's cross-reactive and related but not pathogenic in the artificially inoculated host. And later on, because these there were, the vaccines are apparently not as good as they would like them to be, or there's still more development. They took a regular Merrick's disease virus and attenuated it by passage in cultures. So now they have at least 3 different vaccines around, and my understanding is that these are sometimes used in combination.
That's right.
We can talk about that down the road.
So these vaccines prevent disease in chickens, but they do not prevent the virus from replicating in the chicken and spreading, presumably. And so they say this is the perfect situation to test whether a vaccinated chicken could allow passage of a virulent virus. Now, at the same time, this virus in nature has apparently become more virulent. And starting in the 50s, so originally the virus caused paralysis in chickens when it was first discovered in the 20s or 30s. And then in the 50s, it became oncogenic. And then after that, it started to become lethal. And so you no longer saw tumors because the chickens would die. And so this has been evolving over time. And this is one thing I want to point out, is that the evolution of this virus, the change of virulence, actually predated the use of vaccines. So the hypothesis that the authors put forth in this is that it's possible that the use of these permissive vaccines allowed for the evolution of more virulent strains, but it seems to me that this has happened even before the vaccines were used.
If I'm not mistaken, the niche that we're talking about is farmed chickens.
Farmed chickens, exactly.
Right? We're not. When you say evolution in nature,
I mean farms.
Qualified.
Yeah,
farms.
I guess nature. Right. Farm is not nature, huh? Yeah, maybe you're right, but farmers may not like that. Yes, farm chickens. Now, I call these permissive vaccines, that they allow replication but not disease. They call them leaky or imperfect. And again, I want to object to the use of those words that imply a defect, because these vaccines prevent disease. By the way,
I was wondering what you were going to come up with as an alternative, because any objection, I think you're obliged to come up with an alternative.
So I come up with permissive vaccine.
Okay.
Now, Rich Condit, can you name a permissive human vaccine?
Yeah, I couldn't do it without knowing you and doing TWiV. But don't tell me. The IPV, right?
Bingo.
Okay, I had to think about that because there's IPV and there's OPV, and I have to kind of reason my way through this. But with the inactivated poliovirus vaccine, which is injected,
right,
you get an IgG response, which will prevent the systemic spread of infection with wild-type polio, but doesn't prevent the replication of the virus in the gut where it normally wants to replicate, and it's shedding. Did I get it right?
That's right. It's absolutely right.
Okay, good.
So we have a permissive vaccine in humans, and guess what? But virulence of polio has not changed in all the years we have used this vaccine.
Right.
So this is a natural experiment, in a way, to test this hypothesis, which we already have the data for. And in fact, they don't even mention in this paper, curiously, I don't know, perhaps because it doesn't fit with their ideas, but I just think that, I'm kind of preempting the end of this. We haven't even gotten into the data, but I just think that this whole idea is not supported by any human data that we have. All right, anyway, back to the paper. So they test this by taking chickens that are either immunized or not with a Merrick's vaccine, and they infect them with a range of Merrick's isolates from not virulent to very virulent. And by the way, these viruses they're inhaled by the chickens in the chicken coops. The viruses are shed by the feather follicles, right? And then they get mixed with dust in the coops, and then the birds inhale them and they get infected. So they infect the chickens with a range of these viruses, and then they have these chickens housed in isolators in the laboratory. So this is all done in the lab, not in a farm. Okay.
Yes, but it's quite an experimental setup. I can't.
Oh yeah,
I can't imagine. It's quite an elaborate and laborious set of experiments.
Oh yeah, no, there's no doubt that this is a good effort here,
right?
And these are rather large animals and so forth. But as I said, the experiments are interesting. So they take these chickens in their isolators, they infect them, and they collect dust, and they measure the concentration of virus by PCR in the dust. Okay,
and the way they do that, because I'm always interested in the methods.
Yeah.
I had to look this up. So these, I don't know what the pens look like, but they must be enclosed to some extent because they're ventilated.
Right.
Okay, so there's some sort of a fan that draws air through these things, and there's a filter. So the dust that rises out of these pens collects on these filters, and they daily change the filters, and they come in and they take out the old filter and put on a new one, and they take out the old filter and they shake the dust off into some sort of collector, and then they do PCR on the dust.
Right. And they do an experiment to show that the virus in the dust is actually representative of the virus that's in the feather follicle. I keep saying hair follicle, but feathers. I don't have feathers, but I don't have hair either, but okay, so they do an experiment to show that, which is good. All right, so if you take an unvaccinated bird, you infect it with all these viruses. The most virulent viruses kill them in less than a week, and therefore they're not very infectious for very long because they die, and they don't produce a lot of virus. Now, here's another objection they have. They say during that week, barely any virus was shed. I just really object to the use of barely. I would have said, give me a number. 2 logs of virus or something like that, virus genomes, but they say barely.
This is, you're talking about during the week in the unvaccinated birds with the virulent virus, okay? Yeah. Well, I would object to that as well, because the number that they emphasize here is the cumulative amount of virus over the, 56 days, however many weeks that is, that they do the experiment. And since the birds infected with the virulent virus die within a week, I mean, this is the whole point. You don't have the rest of that time to shed virus. But during that week, they're shedding a ton of virus, okay? It's just that they die, and then it doesn't happen anymore. So you quit collecting data from them. I mean, that's their point in a way, but it's important to word it carefully, because during that week, those infected birds are shedding a lot of virus.
Right, and remember that they're infecting these birds with a number of different isolates of different virulence, okay? Mm-hmm. There is no evolution going on in here. They're not looking at evolution. They're just putting in different virulence levels and asking whether, immunized bird can transmit them longer.
Well, that's, anticipating the bottom line here. That's the biggest problem I had with the paper and with the news bits. Right. Is that the news bits in particular I did not misread, and maybe I was prejudiced by those so that I misread some of the paper. But once again, you got to be very careful with wording, because I was given the impression from a first glance at the paper and the reading the news that they were talking about actual evolution of viruses during the experiment. So when I first looked at the paper, I was looking, okay, if you're going to talk about that, that means you have to see sequence evolution in these viruses during the course of the experiments. I didn't see any sequencing experiments, and it turns out in the end, there's no evolution or any evidence for any evolution. Wasn't even assayed of these viruses during the experiments. It's just a matter of that the vaccine creates an opportunity for prolonged transmission of whatever virus, be it virulent or avirulent.
That's correct.
But, prolonged transmission. But I mean, the virulence things comes in because under normal circumstances, the virulent viruses kill the chickens, so that interrupts the transmission.
Yeah. Now, if you look at the newspaper headlines, imperfect or leaky vaccines can allow the evolution or this more virulent viruses to emerge. That's the conclusion. That's just not the conclusion of
that's
this paper.
Not right. That is not right.
Right. So this first experiment, vaccinated chickens survive these virulent viruses and they shed longer. That's the basic conclusion from that. So the next question is, can they transmit the infection to other chickens? So they do the same experiment. They inoculate vaccinated or unvaccinated birds with avirulent or virulent viruses, and then they put them in a cage with a sentinel bird, which is not immunized. Okay? So you take an unvaccinated bird, you infect it with a lethal strain, they're dead within 10 days, and none of the sentinels get infected. That's the result, although I'm a little surprised because 10 days is a long time.
Yeah.
Anyway, none of the sentinels get infected, none of them dies. Now, if you infect a vaccinated bird, they survive for 30 days, and we know they shed virus. And in that case, all the sentinel birds get infected and die as a result of Merrick's disease. So this shows that the vaccinated birds can carry the virulent virus long enough to shed enough to infect sentinels.
Right. All consistent with their hypothesis. And the experiment supports the data. It's a nice experiment.
Right. And the last experiment really addresses whether chicks born to mothers who are immunized would have enough antibody in them such that if they were infected with a virulent virus, could they then survive and shed? So they take immunized hens, they take their babies, the chicks, and then they infect those. And these comes from either immunized or non-immunized hens. And they basically show that the chicks can be infected with a virulent virus, can shed it for a long period of time, and infect sentinel chickens as well. So the transmission of the virulent viruses not only occurs in the vaccinated chickens, but in their offspring, because the mother passes antibody to the offspring.
And this is relevant to the, in quotes, natural situation, or the situation on a chicken farm, because as they point out, all of the egg-laying hens are going to have been vaccinated. So there will be maternal antibody in the chicks that show up. So this experiment is intended to more closely mimic what's happening on the farms.
Right. So they conclude that all these data are consistent with the imperfect vaccine hypothesis, but I would say they do not confirm it at all, because we do not know why Merrick's has increased in virulence over the years from these studies. There's no experiment here that addresses that. And as I said, I think the virulence has increased before even vaccination occurred. If you wanted to know I think an experiment to do if you want to know whether a permissive vaccine can allow evolution of more virulent strains, you take a vaccinated chicken, you infect it with an avirulent or low virulence virus, you collect what's shed, and then you infect another vaccinated chicken. You keep doing that, sort of like the ferret H5N1 experiment. Mm-hmm. Then you see if virulence increases. Mm-hmm. But you know what, Rich? That would be a gain-of-function experiment.
Can't do that.
Can't do it. But even that experiment, I would argue, is not all that relevant, because it's a laboratory experiment where the situation, the selective pressures simply aren't the same as on a farm somewhere. So in the end, we don't know why these viruses, the Marek viruses, have increased in virulence. They say this could account for it, but I just am not sure.
I think in the field of sort of virus evolution and epidemiology, the term, and I sent you a link to Adam Lowring's Pearl's article on this. We might even want to stick this in the show notes.
Yes, we will. It's a good article.
It's a nice article that explores the relation, really what is the definition of virulence and what is the relationship between virulence and transmission. And this doesn't necessarily give a whole lot of new insight into that, but it does provide one new sort of data point that has been a hypothetical consideration for a long time, which shows that if you, through vaccination, prolong the life of a susceptible host, you can influence the transmission of a virulent strain of virus. That's what it says.
It does not say whether those conditions would lead to the selection of highly virulent strains.
Correct.
Right. So that's what I think much of the press did not pick up on,
Yes.
and it got in trouble here.
Yeah, so as I was reading the Science News piece and the article, and I noted them down here, several statements came out that stuck in my craw. One, the experiments, this is from the news releases, the experiments support the idea that vaccines helped make Merrick's disease deadlier.
No.
I disagree with that. Some vaccines could lead to enhanced virulence. No. Because of these vaccines, the industry has created super hot strains.
Wow.
No.
Wow. Where is that from? It's from some news article.
That's from one of the news. I think that's the Science News article. Opening sentence of the abstract. Could some vaccines drive the evolution of more virulent pathogens? The paper doesn't really address that question. From the paper, we show experimentally that immunization of chickens against Merrick's disease virus enhances the fitness of more virulent strains. Now that's complicated because I think of fitness as a genetic characteristic of the virus.
Yes.
Okay?
Yes.
Though fitness is a complex thing, and I probably don't have enough understanding of this, and some of it is described in Adam's article. But I would be careful with that one, because what's not happening here is actually genetic evolution of the strains. Another one, our data show that anti-disease vaccines that do not prevent transmission can create conditions that promote the emergence of pathogen strains that cause more severe disease in unvaccinated hosts. No.
Have not shown that.
However, in the very end, they do say, our data do not demonstrate that vaccination was responsible for the evolution of hyperpathogenic strains of Merrick's disease viruses, and we may never know for sure why they evolved in the first place. So they do have a correct conclusion there in the very end, but it strikes me as inconsistent with a lot of their other statements that lead to the impression that there's evolution going on in the experiment. So,
Yeah, I mean, I think this could have been written in a much better way than it has been.
Well, this is one of the problems I have with science in general now. There's a lot of pressure to sell yourself. I suppose there's always been this kind of stuff, but to me, science, it's a pure art form. The investigator should not be part of it. The salesmanship should not be part of it. It should just be the experiments. Yep. But I guess I'm a dreamer.
I want to hit one last point in their discussion. They make the statement, quote, whether leaky human vaccines could also create the conditions in which more virulent strains can thrive will depend, among other things, on the selective factors currently preventing the emergence of hyperpathogenic strains in human populations, end quote. So that comes back to what I said at the beginning of our discussion here. We don't know what are the selective pressures that govern the evolution of viruses in most cases. We know that antibody does. We know that antivirals do if we use them. But that's one of many, many others. And so the idea that just allowing replication by having a vaccinated host would lead to this, I think, is oversimplistic. Let's just take a hypothetical. Why do hyperpathogenic viruses have to emerge anyway? Why? What's the point? They transmit pretty damn well as it is, over the range of lethality that we see in viruses. They're there today because they can transmit and survive in human populations. So I don't understand. Epidemiologists and basic virologists together, they have this fascination with the evolution of virulence, which is the subject of that Loring paper, the pearls, right?
Right.
And I just do not understand that because, as far as I can tell, it doesn't change all that much, at least in human viruses. So I think we have no clue about what kinds of selective pressures are at play here. I think, just to finish up, they say that our data don't demonstrate that. Our data show that. Wait, sorry, I'm screwing up here. Whether leaky human vaccines could create the conditions that their sentence that I quoted, they should have quoted the Salk poliovirus vaccine situation, which clearly speaks against this notion because the virus has not evolved when we're using a vaccine that does allow replication.
Well, to me, this to some extent, that sort of a statement is a way of raising the idea that maybe this does go on in nature without actually saying it because your data doesn't support it.
Yeah,
it's like it's the Glenn Beck, Rush Limbaugh approach of saying, as I said on another TWiV, now I'm not saying that Condit is a jerk, but some people think,
Yeah, right. That's right. That was great. So Ed Young, who sent me this article, he asked me, do you think this has any implication for the development of any kind of animal vaccine? And I said, no, I don't think so. I don't think this provides any evidence that this hypothesis is right. It shows that a virulent virus will go through a vaccinated animal and get transmitted. It's kind of intuitive that would happen. It says nothing about evolution and selective forces. So we have IPV, which allows replication. It's a great vaccine. We're going to be using it for another 10 years. So...
And I think that there's, what the paper does is that there is this ongoing discussion about the relationship between virulence and transmission and the variables that go into both virulence and transmission. And this is an experiment that addresses to some extent that relationship, and that'll be of value in the future. Yep. Actually, the one thing we didn't discuss is that there is one of the things they cite is the classic case of attenuation of virulence in a fairly natural situation, and that was the introduction of Mixoma for control of rabbits in Australia. And the situation, we've talked about this before. Australia was overrun with rabbits. They introduced a lethal rabbit pox virus, Mixoma, and within the first few years it wiped out 99% of the rabbits. But then the rabbits became more resistant and the virus more attenuated until it reached a new equilibrium, and the rabbit population came back. Now, one of the things he says in this is that there was, following that, an uptick in the virulence of the Mixoma virus. Now, I'm not familiar with that. I would have to read that, but that's a new one to me. At any rate, that's a situation in nature that is relevant to this whole thing.
The Mixoma story is very interesting because it's really complicated. Mm-hmm. And measuring the virulence of the isolates that they pulled out of rabbits, in that first year and in subsequent years was not straightforward. In fact, they had to put them into different classes of virulence depending on how many rabbits were killed, It's not simply a matter of decreasing virulence, I think, and maybe at some point we could do an entire TWiV on that whole story, because Fenner spent years studying these
yes, right? It would be good to do that and do that properly, do it well.
Right.
So the other thing I noticed out of this paper, this is sort of a sidelight, but this is something that fascinates me. I described the vaccines that were used in the beginning of this discussion, and they I noticed reading up on this in a Wikipedia posting that current vaccines use a combination of vaccines consisting of HVT, basically consisting of a mixture of the turkey viruses and the attenuated Merrick's disease virus strain. And this reminded me of, and you're right, Vincent, I'm pretty sure I'm positive we discussed this earlier. But there was a situation that arose in Australia where immunization against a different poultry herpes virus, and I forget what it's called, but it's a lethal herpes virus of an alpha herpes virus infection of poultry. And there were two different attenuated vaccines created that were being used in the same populations of chickens, and they recombined with each other because they did the genetics of their attenuation was different, so that recombinance between the two vaccine strains could regenerate a virulent virus, and that wound up spreading virulent disease through these flocks, and I believe even being established outside farms. So I'm not 100% sure of that. So when I read this thing about using combinations of different herpes viruses in vaccinating these chickens, it reminded me of that, and I thought, geez, that's a whole lot scarier than in a way using a vaccine. What do you call them? Not imperfect, not leaky.
Permissive.
Permissive vaccines.
Yeah.
At any rate.
So it's funny, this paper on the recombination of these vaccines leading to a more virulent virus that was also sent to me by Ed Young about a year or so ago for my comment. Yeah, so Ed is on this. He's interested. By the way, Ed Young, great writer, writes for National Geographic. He's got a blog there at the moment and is moving on, I think, to the Atlantic.
Where is he located?
I will give you a link right now. Ed Young can be found at, so the name of his blog is Not Exactly Rocket Science.
Okay.
It's phenomena.nationalgeographic.com. It's / not exactly rocket science.
Where is he physically located?
He's physically in the UK.
Because there's somebody else I'm blanking on his name, Ed in Edinburgh or Glasgow, who's interested in virus evolution. He's even published with Reed. This is terrible. I'll find it. As a matter of fact, I'll find it.
Don't remember. Okay.
I can't find it right now. I'll come across it later on.
Let's do a couple of email.
Sure.
Yeah, we can. It's just you and me, right? We're in no hurry.
Yeah, we can do this.
Let's do a couple. Can you take Gerald's?
Sure. Gerald writes, July 2015, I was watching TV and saw this slick ad against vaccine. He didn't provide a link or anything, did he?
No.
So he saw a slick ad against vaccines. I have some firsthand experience with this, quote, smarter than the collective knowledge of 1,000 years, unquote, thinking. Although not related to vaccines, rather antivirals, it smells the same. I think it is problematic to be influencing people on TV regarding public health issues. I wonder how many trained immunologists support that coalition. Probably like one crazy one. I feel as if the internet can allow people to learn just enough to be dangerous. They go to the fountain of knowledge, I love this, and gargle rather than drink. Perhaps this can be an opportunity to educate the public. What does it take to convince people that with vaccines, the benefits outweigh the risks by a large factor? Can animal studies be demonstrative in a way that is condensed to between 5 and 25 minutes? This is about the amount of time one needs to develop a complete understanding of immunology, right? Tongue-in-cheek statements aside, that is all the time people would give, if any at all. Attention spans nowadays are short. Let's hope we don't go back to the old days with lots of bad disease. Let's not raise a nation of adults extra susceptible to a rogue nations of weaponized pathogens. Vaccines work, and I hope we don't let ourselves forget it. Anyhow, just putting in my 2 cents, Jenny. Well,
Jerry.
Jerry. Oh, Jerry. I'm sorry, Jerry. I think the ship is slowly turning on this. I cite the California legislation that is saying you can no longer claim exemption from vaccination for your kid to enter into school. And our experience on the show so far, I think, is that for all the reasons that Jerry cites, it's hard to persuade the public with a lot of numbers and scientists and white coats and that kind of stuff. What convinces them is anecdotes. Mommies whose children have died because they were unvaccinated, individual cases.
Yeah.
So,
I mean, this idea of educating people on TV about public health, I mean, we do that in a positive way all the time, right?
Mm-hmm.
We have many ads to take this, get your vaccines, take this in case of this. I think it's okay, but we know that vaccines work, and there's absolutely no reason for TV stations to allow advertising that says vaccines are not good. This is absolutely wrong. And
right.
What comes to mind is what I heard at ASV. Maybe you heard this, Rich, but the University of Toronto is going to be offering a course called The Dangers of Immunization. And there's been,
yes, that's right.
There has been a huge outcry against this, but the dean has insisted, or maybe the president of the university has insisted that, we need to have both sides of all arguments, so this course will go on. This is one argument where there aren't both sides. There's one side, vaccines work. And so I think this is horrible. The trustees should get rid of this president of the UT or whoever it was that did this. You should know it for a course.
This is where we need video so you can watch me cringe.
It's unbelievable.
It's really bad.
I just don't understand. Now, unfortunately, we have another email about this. And actually, we have two more.
Excuse me, I want to interrupt for a second.
Yeah.
It's Eddie Holmes. Okay?
Oh, yeah, Eddie Holmes. He's in Australia now. He moved.
Okay. But he was in the UK,
was he? He was, and then he went to Penn State for a while.
Okay.
And now he went to Australia. Right.
Part of the communication with Adam Lowering, I had some correspondence with him on this, is that we can perhaps anticipate more about the evolution of Mixoma from maybe Reed and Eddie Holmes, which would be nice. That would be great. Okay.
All right, we have two more emails about vaccines. The next one is from DJ, who writes, I've been researching immunization vaccination papers for at least several years and have developed a complex opinion as to the scientific validity of this mass medical practice. All right. This letter goes on for a bit more, but the only opinion about this mass medical practice is that it works. It saves lives. It makes us live longer. There's no question. Sometimes there are side effects. As we said last time, sometimes a vaccine may induce narcolepsy in a small number of individuals, but in the vast majority of people, it prevents you from getting an infectious disease. And the benefit far outweighs any negatives.
I think the thrust of this letter in the end is that the author, DJ, senses what he calls a staidness, perhaps a dogmatism,
right,
among the scientists, because we are so forceful in advocating the benefits of vaccines that the impression can come across that we're closed-minded about the some of the potential safety issues. And he goes on about the narcolepsy thing, which in fact we covered. Right. And I think it's important to understand that, we feel strongly about the use of vaccines, but not to the point where we're unwilling to discuss or take into account some of the potential adverse effects that can happen, and and deal with them. One of the unfortunate things about vaccines, and the narcolepsy case is a good example of this, and the author of this letter alludes to this, is that even in a large phase 3 trial, you can't necessarily uncover all of the possible side effects that may be very rare side effects that happen in a small portion of the population that happens to be genetically susceptible. And it's not until you put the vaccine out in the population and vaccinate hundreds of thousands of people in many different areas and many different age groups and et cetera, that some of these rarer complications might come up. I'm sorry, you got to do it, okay? And when those things come up, you take note. We keep track of adverse effects. And when they crop up, people take note, they study it scientifically, they figure out what's wrong, and they make the appropriate improvements. That's the way it's done.
Yeah, the narcolepsy is a great example of that.
Great example.
And ended up with a science paper that says this is what's going on.
Right.
So DJ says, I'm not... he said, there's no doubt that targeting certain pathogens is a tremendous gain, but he, as Rich said, he just objects to saying, we should use this all the time. And I think Rich's response was great. Let me get down to the end of his letter. You can read his whole letter, by the way, at twiv.tv/twiv-348-letters. I'm not advocating a moratorium on vaccines or on their development. However, I am attempting to avoid the dogmatic staidness sometimes found in the academy by suggesting a more open dialogue and further directed research. So this is what Rich just alluded to. You make your vaccine, you have to deploy it in lots of people before you know if it's good or not. As with any good science, hypothetical deduction, detailed empirical exploration, and reasoned dialectical discourse should enlighten and inform any mean ephemeral progress. I don't think we disagree.
No, and I think we do that. I think we do that.
Yes.
I think what's confusing is that we can be very adamant about the use of vaccines, basically in response to all the anti-vaccine stuff that goes on, and that can be mistaken for a really dogmatic perspective on it.
Right.
I didn't understand until TWiV. And looking into this stuff, I really didn't understand this concept that you can't really know ultimately how safe a vaccine is going to be until you market the thing.
Of course.
And then that's just the way it is. You can't do, because of the cost constraints and just practicalities, you can't do a phase 3 trial that will ensure absolute safety of a vaccine before marketing the thing. There are risks to this, but as was said, I think, in the previous letter, it's pardon me, because we have discussions about this as well, but it's a risk-benefit situation. The benefits far outweigh the risks.
Look, you identify a medical need. Right now, we've identified Ebola viruses as a medical need. We're developing vaccines. Do you think anyone in at-risk populations in Africa, when we present them with a phase 3 tested, approved vaccine, will say, no, I want to wait till you put that into millions of people to see if there are any side effects before I take it? No, because they are at risk for catching a lethal disease. Now, if it does have a side effect in millions of people, we'll learn about it. Phase 3 will get you through the major ones. The narcolepsy was a very rare thing that only cropped up with large numbers in a certain population of individuals who are genetically at risk.
And in saying that it was rare and that kind of stuff, we're not brushing it aside, okay? People got hurt. That's not good. But the science paper is terrific because it tells us why they got hurt and provides a tremendous amount of insight into that, and it tells us how to make the appropriate changes so that people don't get hurt in the future, not only with respect to that influenza vaccine, but potentially other vaccines as well.
All right, the next letter is from Pete, who writes, I'm slowly but surely catching up with all the TWiV that is out there, sating my daily fix, though I am a bit concerned when I eventually do catch up and have to convert to a weekly dose. And he sends a link to a paper on chronic fatigue syndrome, which we actually covered on TWiV 345.
Right.
So you must have run into that, Pete. Cape Town is currently 8 degrees C, 87% humidity. Wow, which is unusual and quite sunny for a winter's day. It's humid for a winter's day.
8 degrees C. I have to think about that. That's
very cold.
That's very cold.
Yeah, but the humidity is huge, right?
Yeah.
Wow.
Right.
Yeah, this was the paper we discussed showing that wiping out B cells with a monoclonal can relieve symptoms in people with chronic fatigue syndrome.
Right.
Take the next one, Rich, please, Mark.
Mark writes, Dear TWiV, keep up your educational outreach and evangelizing against the stupidity of the anti-vaxxers out there. Fortunately, here in California, our governor recently signed a law tightening vaccination requirements for school children by eliminating personal and religious beliefs at exemptors from vaccination. And he gives a link to a news release on that law. The fight against anti-vaxxers is truly nationwide. Via LinkedIn, I saw this post from a classmate who is the chair of pediatrics in a department at Dell Medical School in Austin, Texas, and he gives another news link to an article out of Texas that describes how some practitioners, and we've covered this before as well, some practitioners, pediatricians, are refusing to take as patients children who are unvaccinated because of the potential for them to bring disease into the clinic and spread it through the clinic to other children. And so that's a really strong measure that I support. This article was from a pediatrician that supported the practice as well. Today's high temperature here in San Jose, California was 77 degrees Fahrenheit and mostly sunny. As I write this, my watch informs me that the outside temperature is 70 degrees Fahrenheit. Here is an easy-to-follow fitness tip for TWiV-AD listeners. Group your return phone calls together. Make them in a block of time and take a walk while chatting on the phone. Urban street noise isn't a deterrent. The counterparty will respect your walking, talking. It's easy to accumulate 1 to 2 or more miles walking and talking instead of sitting. All the best, Mark. Good idea. All right. And we got fitness ideas from our TWiV listeners. That's good. TWiV is good. TWiV is good for you.
It is.
In more good for your body as well as your mind.
All right, one more from William, who writes, Hello, esteemed TWiV hosts. I am very excited to finally write in. I've never really had a good reason before, but now seemed like a good time to speak up. I discovered your podcast while looking for podcasts about 2 years ago now and have been a weekly plus listener ever since. I love the conversational tone and the pragmatic attitude that you bring to the table, i.e. science is done by people, for people, and must always be considered in this light. I work for a nonprofit ed tech company in Silicon Valley where we make free educational materials for K-12 students.
Good for you.
The weather here in California is too hot and too dry, partly cloudy, 66 Fahrenheit, 18 C, with heavier winds in the afternoon these days. At
66 Fahrenheit is too hot?
Really? Too hot, at least to my liking. I'm writing because I would like to start a weekly ed tech podcast and would love to use the title TWiET, TWiET, This Week in Education Technology, if that's okay with you. I also manage a bug-infested apartment complex, so if you know of any viruses of arthropods that could bring about a cockroach apocalypse without pesticides, I would love to hear about them. Besides that, I just want to say that I love the podcast, love you guys, and I hope you never give it up. Keep up the great work.
I'll tell you, if you could find a virus that would cause a cockroach apocalypse, you could make a lot of money. So go for it.
Do you think it would evolve to lower virulence?
Yeah, that's a problem. That's a problem. Would the cockroaches become resistant?
Maybe. They're resistant to everything else already, right?
Yeah, they're tough.
Of course, it's fine to use This Week in Education Technology. It's not patented by anyone. Go for it. Good luck. And if anyone knows of viruses that infect cockroaches, let us know. I'm not aware of any, but I know there are, but not whether they would be good for pest control. Hey, Rich, let's do some picks.
Good idea.
What do you got for us today?
Okay, so matter of fact, I need to text you a photo here. So I said early on that I was podcasting from Sun River, Oregon, from a family house. And so this is a episode relevant pick. The patriarch of this family is one HJC, a.k.a. Jeremy Swan, who some of our clinically oriented listeners, that may be a familiar name. I may have even talked about this on TWiV before. He was the inventor of a device called the Swan-Ganz catheter, which is what's called a flotation catheter. And my picks are to a wiki article on aortal catheterization that talks about this, and a video about how the Swan-Ganz catheter works. And it's quite an important medical development. Prior to this, people had done heart catheterizations, where you stick a tube in an artery and run it up in the heart to either inject things or do various measurements and that kind of stuff. And prior to this invention, that was a difficult procedure to do because it was hard to direct the catheter to the right place, and the tip of it, if it poked against the wall of the heart or something like that, could cause fibrillations. So it was a difficult procedure to do and cause problems. And what my father-in-law invented was the fairly simple concept of a catheter that had a little balloon on the end of it that could be inflated with air when you stick it into the artery initially. You stick this thing into the artery and inject a little air into it, and the balloon floats in the bloodstream. And I remember him describing to me the first time he used this thing, he was absolutely astonished because the bloodstream just sucked this thing in. Okay. Mm-hmm. And so it guides the thing because it floats in the vessel, and it also gives it a soft tip so that it no longer causes the fibrillation. And if you look at the cartoons, or look at the video, there's a whole bunch of other modifications that are done to this that allow you to do injections and measure pressures and all the rest. So that was a significant scientific advance in cardiology, and I want to acknowledge my father-in-law. I'm sitting in his office in his vacation home looking. I just sent you the picture of a big thing that celebrates all of the fellows that studied under him. There must be hundreds here. While he was the chair of cardiology at Cedars-Sinai.
So this is your wife's father?
My wife's father.
Cool.
Her maiden name is Elizabeth Pamela Swan.
Wow, cool. Yeah, I know you had mentioned this before, but I didn't know, for some reason I didn't know it was your wife's dad. I thought it was your daughter's father, one of your offspring's husband or wife's father, for some reason.
No, it's my father-in-law.
Cool.
Who, he is no longer with us.
What year did he develop this? Do you know?
I believe it was 1970.
So my dad used to do this procedure because he was a
is that right?
He was a thoracic surgeon. He used to do this.
Okay.
Yeah, yep. He used to talk about.
There you go.
Yep.
Yeah,
cool.
I mean, this guy's a verb. Okay, you swan people. Okay.
This is wonderful.
I wonder if anybody is ever going to get Racaniello'd.
I doubt it. Unless you listen to TWiV, that's being TWiV'd because it's not just me. That's okay. That's great. Very cool. And it's a nice picture. And you're sitting in his office.
I'm sitting in his office. I probably ought to take a selfie of that.
Because he, this thing that you're in is like a family vacation place, right?
Yeah, when he built this place, part of the build was a little office for him in the place. I don't know how much he used it, but he's the kind of guy who never sort of completely quit working.
Yeah.
So.
All right. Here very cool.
I can take I'll get a selfie of this for you.
All right, while you're doing that, my pick is something that's not going to be for everybody. I usually don't pick hardcore science, right? But today I want to pick a book called Mobile DNA 3. And it is a book published by ASM Press, which I received not too long ago. And I love mobile DNA. This is a brand new edition of a multi-author book all about DNAs that jump around. 40% of our genome is made of DNA that got there by jumping around. And these jumping DNAs have specific mechanisms, transposons, transposable elements, lines, LTR transposons. There are tons of them. This book is over 1,300 pages.
Holy cow.
Must weigh 10 pounds. It is my summer reading. I love mobile DNA.
You're going to read this thing?
I can read the whole thing, but I'm going to bone up on mobile DNA because I find it really interesting. These things contribute to evolution. They're really important, not just of cells, but of viruses also. Let me just tell you about one of my favorite mobile elements. It's called Sleeping Beauty. Ever heard of Sleeping Beauty, Rich?
I have heard of Sleeping Beauty, but tell me about it because I can't keep these things straight. I taught a course in transposons probably over 30 years ago. I was still in Buffalo, and it was a gas, but so much has happened since then, and I haven't kept up with it.
Well, Sleeping Beauty was a transposable element first discovered in a fish genome. All right? So a transposable element is something that moves from either within the genome or somewhere else by many different mechanisms. And Sleeping Beauty was found in a fish genome, but it was extensively mutated, so it wasn't active. It couldn't move around. So they corrected all the mutations and made something that would now transpose. And that's what they called it. That's what gain of function, exactly. And this was done ages ago. And that's why it was called, because in the fish genome, it's sleeping, right? And they fixed it and it woke up, so that's why they call it Sleeping Beauty. Okay. And now this thing has been studied extensively. The proteins. The thing is a DNA that encodes what's called a transposase, an enzyme that helps it move around. And then the ends are direct repeats, inverted direct repeats that go into the genome. That's just one example. That's one chapter. I love it. I know it's not for everyone. It's a huge book. It's I think on Amazon you can get it for $140, but I know you're not going to buy it. But I just am very excited about transposable elements. When this showed up in my mailbox, actually on my front step because it's too big for a mailbox, I just
Is the book stable? Do the pages stay in order? Do they jump around?
That would be nice, wouldn't it? That would be really nice. But it covers transposons in eukaryotes, prokaryotes, viruses, cells. Really cool. Multi-author, editor-in-chief Nancy Craig. And if you are into transposons, you should check it out. We have a listener pick from Kevin who wrote, is it considered gain-of-function research if you make a synthetic prion? So this is a paper in PLOS Pathogens. Intraperitoneal infection of wild-type mice with synthetically generated mammalian prions. So they synthesized the prion protein, and they can infect mice. And so that's a good question. Sure, you've given it a function, right?
I guess. You've created function.
Yeah.
But the interesting thing about this paper is that, as they point out at the end, it absolutely and once and for all, if anybody had any remaining doubt, nails down the notion that prions are protein, period. Mm-hmm. Because you can fabricate this protein from scratch, and it has all the properties of a prion.
Yeah. So they make them in vitro by a specific way, but it is quite clear, yeah. Well, thanks for that. And that will do it for TWiV 348.
Jeez, we have to stop? Can't we keep going?
Yeah, I think 2 hours.
How long have we been at this?
For 2 hours.
2 hours.
All right. But, our listeners, they need to go do other things. I had a good time.
What else is there to do?
Yeah.
I don't know.
348, you can find it at iTunes, twiv.tv. And you can also get it on your mobile device, whether it be an Android or an iPhone. You can get a pod-catching app and download it, subscribe for free. There are apps for both platforms that you can use for that. And if you have questions or comments, we really enjoy hearing them. TWiV at twiv.tv. Even if you want to just say hi, we love your podcast, or we don't, or you should fix something, or you have a question, just send it. We love it. We like contacting you guys out there. We like knowing who you are and how long you've been listening, how you found us, and so forth. So
yeah, I think the email is one of the best parts. It's always I love doing it. I feel like we're kind of the Dear Abby of virology or something like that. It's great stuff.
Rich Condit is what am I supposed to say? University of Florida, Gainesville.
Yeah, sure. University of Florida.
What was your title? You're a
emeritus professor.
Yes, Rich Condit is emeritus professor at the University of Florida at Gainesville. Thank you, Rich.
Sure enough, always a good time. I love doing this.
I'm Vincent Racaniello. You can find me at virology.ws. You can also find me on Twitter, PROFVRR. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.